Here are the goals for the Cellular Respiration Lab chapter. Once you have completed this chapter, you should have met these goals. In cellular respiration, energy is transferred in a series of redox reactions. Redox stands for reduction oxidation. These reactions involve the transfer of electrons from one reactant in a chemical reaction to another reactant in that same reaction. One mnemonic device you can use to remember what redox reactions involve is LEO goes GR. In this mnemonic, LEO stands for loss of electrons is oxidation, while GR stands for gain of electrons is reduction. Adenosine triphosphate, or ATP, is the main energy storage molecule in cells. Energy stored in ATP can be transferred to various endergonic reactions and processes in cells. The process of cellular respiration transfers the energy in glucose to ATP molecules. Examine the chemical equation for cellular respiration. Take a moment to recognize what the reactants and the products are in this reaction. Cellular respiration includes four processes: glycolysis, pyruvate processing, also known as the formation of acetyl-CoA, the citric acid cycle, also called the Krebs cycle, and oxidative phosphorylation, which includes the electron transport chain and chemiosmosis. Glycolysis is the process by which glucose is split into two pyruvate molecules. Here you see a chemical equation for glycolysis. Note that this is a reduced equation. This version does not include the two ATP molecules that are spent during the energy investment phase of glycolysis, nor does it include the total of 4 ATP molecules being produced during the energy harvesting phase. Recall that in total, glycolysis will spend 2 ATP, but produce a total of 4 ATP for an overall net of 2 ATP molecules produced by glycolysis. The carbon dioxide produced in cellular respiration comes directly from the carbons in the glucose molecule. Those 6 carbons in glucose provide the carbons necessary for the 6 carbon dioxide molecules. We can measure the rate of cellular respiration by measuring the amount of carbon dioxide produced. But carbon dioxide is a gas, so how can we test for carbon dioxide? When carbon dioxide is combined with water, it produces carbonic acid, as shown in this chemical equation. Bromothymol blue is an indicator of pH. It maintains its blue color in neutral and basic pHs. However, it turns from blue to green to yellow in increasingly acidic conditions. You can follow along with this activity in your manual. This activity compares the carbon dioxide in atmospheric air versus the air exhaled by an animal, specifically a human. The initial setup is to add water and bromothymol blue to 2 separate test tubes. For atmospheric air, a straw is inserted into the solution in one tube. A bulb is then used to pump air into that test tube for approximately 2 minutes. For the human test tube, a person uses a separate sterile straw to exhale into the second tube for the same amount of time that atmospheric air was pumped into the first tube. This slide shows the initial and final results from the experiment. Take a moment to record the initial color and the final color as the results in the table in your manual. In the next experiment, we compare cellular respiration in animals and plants. For animals, this time we will use beetle larvae. For plants, we use germinating peas. Both organisms are alive and performing cellular respiration. Follow along with this activity in your manual and take notes as you go. Here we are going to determine what our independent and dependent variables are. The dependent variable is dependent upon the independent variable. The dependent variable is also what we are measuring. So what are we measuring here? Right, the production of carbon dioxide. So what is the carbon dioxide dependent upon? Also right, the organism that is respiring. So what is your prediction? Which will respire more, plants or animals? Our procedure is to acquire 3 test tubes and add 5 drops of bromothymol blue to each. Remember, the bromothymol blue is going to be our indicator of carbon dioxide production as it changes color. To each tube, we add enough glass beads to have a dry layer on the top. On top of this, we add a single folded piece of cotton gauze. We then add 8 to 10 more glass beads. These materials are used to provide a platform on which our organisms will be placed. We now add 8 beetle larvae to one tube, 4 germinating peas to another tube, and nothing to the last tube. This tube will be our control. The reason we add 8 larvae but only 4 peas is because the peas have about double the mass of the larvae. So by adding twice as many larvae, we have approximately the same mass of each organism in each tube. We then add a cotton ball to each tube to help trap the carbon dioxide in the lower part of the tube, and then seal each tube with a cork. This photo shows the experimental setup for each tube. Feel free to pause here and examine the experimental design more closely. The initial color for each tube is noted in the data table. Each tube is allowed to sit for up to 15 minutes before the final color is noted in the table. During the 15 minutes, the organisms are respiring and the bromothymol blue should change color as a result. These photos show the initial color of the bromothymol blue in each test tube before the experiment begins. Record the initial colors in the data table. These photos show the final color of the bromothymol blue in each tube after the experiment has finished. Record the final colors in the data table. As you can see, we see evidence of cellular respiration in 2 of the 3 test tubes. Does this make sense? Yes! Our living organisms respired and produced carbon dioxide, resulting in the color changes we see. This is the end of the activities for this lab. Be sure to go back through all pre-lab and post-lab questions, as well as questions throughout the chapter to ensure you understand the concepts and procedures.